One molecule, three effects, no genetic modification required. The Ashibo engine even predicts the molecular formula, C18H29N5O5. Molecular weight, 395, a standard drug-sized molecule. Let me paint the picture of what this could become. Imagine a farmer in Ethiopia or India or Brazil. They go to their local agricultural supply store. They buy a bottle of Rubisco activator. They spray it on their wheat field, their rice paddy, their teff crop. The molecule penetrates the leaves, enters the chloroplasts, and binds to pocket number 5 on every Rubisco enzyme in every cell. Suddenly, Rubisco makes fewer mistakes. It fixes more CO2. The plant grows faster, no GMO, no regulatory battles, no 15-year development timeline for genetic engineering, no need to breed it into every variety of every crop separately. One molecule, every C3 crop on Earth, wheat, rice, soybeans, potatoes, barley, teff. C3 crops account for 75% of global food production. If a Rubisco activator could improve their photosynthetic efficiency by even 15 to 20%, not the full 60%, just a fraction of it, that would be worth tens of billions of dollars annually and could feed hundreds of millions more people. And we found not just one pocket, we found 4 novel sites. Plus a pocket on the small subunit that nobody knew existed, plus a site that could chemically mimic Rubisco activase, solving the heat sensitivity problem that causes crop losses during heat waves. You might ask, if this is so obvious, why didn't anyone find it before? 3 reasons. First, the mindset barrier. Rubisco researchers think in terms of mutations and genetic engineering. The concept of a Rubisco drug simply didn't exist in their vocabulary. Nobody looked because nobody thought to look. Second, the tool barrier. Conventional pocket-finding software was designed for pharmaceutical targets, kinases, GPCRs, proteases. Nobody thought to run them on a plant enzyme. And even if they did, those tools look for simple concavities. The Ashibo method maps the physics, the actual electromagnetic field landscape, which reveals pockets that geometric methods miss. Third, the disciplinary barrier. Plant science and pharmaceutical science are separate worlds, separate journals, separate conferences, separate funding agencies. The person who understands Rubisco biochemistry doesn't know about druggability analysis. The person who finds drug targets doesn't study photosynthesis. Ashibo biophysics sits at that intersection, and that's where the discovery was waiting. This is not a theoretical exercise. The data is real. The pockets are mapped. The molecular formulas are predicted. What's needed now is experimental validation. A medicinal chemist needs to synthesize the predicted molecules. A plant biochemist needs to test them on Rubisco in vitro. An agronomist needs to spray them on crops and measure the yield difference. We are making the Ashibo biophysics platform available to researchers worldwide. If you are a plant scientist, a medicinal chemist, an agricultural biotechnology researcher, you can analyze any protein on our platform at ashibobiophysics.com. We are particularly reaching out to researchers in Africa, in Ethiopia, Kenya, Nigeria, where crop yield improvement isn't just an economic question. It's a food security question, a survival question. The tools exist. The targets are mapped. The pathway is clear. For 30 years, the world tried to engineer a better Rubisco and failed. Maybe the answer was never to change the enzyme. Maybe the answer was to find the right molecule to help it work better. We found where that molecule needs to go. Now let's build it.